Right. So I'm going to give a bit of a different talk today. I'm not going to tell you about my own research, but I'm going to give you an overview of the research done at the Center for Engineering Mechanobiology, in particular highlight one of the research trusts, which is focused on the nucleus and mechanical memory. So I don't need to convince this audience that mechanics is important for tissue and cell function. Different tissues in our bodies can have vastly different levels of stiffness. And the cells that reside within these tissues sense the stiffness and adapt to it. The stiffness can also change as a result of injury or diseases. And again, the cells have to adapt to that changing environment. And in addition to these more static mechanical cues, there are also, as you heard, dynamic external mechanical forces and cues that act on the cells. And so, as you already heard from Vivek, the Center for Engineering Mechanobiology is a multidisciplinary science and technology center that is focused on engineering mechanointelligence and understanding this dialogue between cells and their external mechanical environments. You already saw this cartoon in Vivek's talk. The center has three different research trusts or IRTs focused on different aspects of this dialogue. These IRTs are not isolated. They're actually highly interconnected and they talk to each other. But today I'm going to focus on the second IRT, which is about nucleus and mechanical memory. And later on during the meeting, you'll hear from Becky and Trina about the other two IRTs. And so before I get into, the IRT and our goals and accomplishments, I want to start with a brief introduction. The nucleus arguably is one of the most important organelles within the cell because it houses the genome, the genetic material who makes us who we are. As you know, the genome is not housed inside the nucleus as just a linear sequence of letters. Human DNA, when stretched end to end, is 2 meters in length. And that 2-meter long DNA has to be folded and compacted to fit inside the space of the nucleus. Thanks to recent advances in sequencing as well as imaging technologies, we have come to appreciate that the genome folding does not happen randomly, and the genome is not just a random ball of spaghetti inside the nucleus, but it's highly compartmentalized. That compartmentalization spans several length scales. So I took this cartoon from a recent review from Giacomo Cavalli's lab in France, which I think really nicely illustrates this multi-scale organization of DNA within the nucleus. I don't know if you can see my cursor. I guess not. Let me just try to change to a laser pointer. So at the smallest length scale, we have the nucleosome where 146 base pairs of DNA wraps around an octamer of histone proteins to form the nucleosome. This is the basic packaging unit of chromatin and the repeating unit of chromatin. And using advanced microscopy methods, my lab showed that nucleosomes fold into heterogeneous and small groups, which we named nucleosome clutches. And we have evidence that suggests that the size and the compaction of these nucleosome clutches is important for underlying accessibility of the DNA and has functional relevance. Now, at the next length scale, multiple nucleosome clutches cluster together to form chromatin nanodomains. The functional significance of these nanodomains are not yet clear, but we know that architectural proteins like cohesin then extrude loops through these chromatinized DNA to form the next level of organization known as topologically associating domains or TADs. And these TADs form architectural units where the DNA segments within an individual TAD tend to interact more frequently with each other than with neighboring TADs. And so again, these have functional relevance and significance. The TADs further organize into A and B compartments, or active and inactive chromatin, or euchromatin and heterochromatin. And then these A and B compartments further organize into chromosome territories within the nucleus, where each chromosome occupies its own territory and does not intermingle with other chromosomes. And so there's multiple levels of compartmentalization, and these different levels of compartmentalization seem to play an important role in regulating transcription and gene activity. Now, our understanding of gene regulation is still incomplete. Again, I took this cartoon from a recent review article that Marcus Woodworth, a postdoc in my lab, wrote together with me. And he made this really beautiful cartoon to illustrate that gene regulation is a big puzzle, and multiple pieces of the puzzle have to come together and fit together for a gene to be activated and repressed. But the multi-scale organization that I showed you in the previous slide actually plays an important role in this puzzle. And so again, architectural proteins like cohesin and CTCF interact with each other to extrude loops and organize chromatin into domains. Loop formation can also be important for regulating enhancer-promoter communication. We have these other domains like nucleosome clutches that compact DNA and make it inaccessible to transcriptional machinery. And this relies on epigenetic modifications, mainly on the histones. And then in addition, we have mechanisms like phase separation, where transcription factors, transcriptional machinery can form hubs. And these hubs have to come together at the right place at the right time, find accessible DNA in order to activate a gene. So this is a very complex, regulatory mechanism, and we don't really fully understand how all these pieces yet fit together. But what we do know is that the mechanical forces from the external environment actually influence all of these mechanisms, because the nucleus is not isolated from its external environment and from the rest of the cell, but it's actually tightly coupled to the external environment. And again, I took this beautiful cartoon from a recent perspective article from Guy Jennings' lab, where you can see very beautifully that on the nuclear membrane, we have the link complex. The link complex connects the actin and the microtubule cytoskeleton to the nucleus. And the actin and microtubule cytoskeleton is also connected through the focal adhesions into the extracellular environment. And so the forces that are acting through the extracellular environment are transmitted through the actin and microtubule cytoskeleton onto the nucleus. The nucleus senses these forces, can be deformed by these forces, and this in turn can change the spatial organization of chromatin and DNA at multiple length scales. In addition, we have these channels on the nuclear membrane, the nuclear pore complexes that regulate nucleocytoplasmic transport. And so mechanosensitive proteins and transcription factors, as well as epigenetic regulators, can flow through the nuclear pore complexes in and out of the nucleus in response to mechanical cues, once again changing the epigenetic and the chromatin landscape of the nucleus. All those changes can then lead to changes at the transcriptional level and rewire the transcriptional programs. And so given this tight link between the external environment and the nuclear architecture and transcription, it is tempting to hypothesize that mechanical memories can be stored within the nucleus in the form of all these different mechanisms that ultimately determine specific transcriptional programs and outputs. So with that introduction, the goal of this IRT is to understand how mechanical memory is developed through changes to the morphology of the nucleus, through changes in the spatial and temporal epigenetic activity, and transcriptional activity. And if you can understand how all these things come together to develop mechanical memories, then one of the cool applications would be that we can then rewire mechanical memories. We can strip unwanted memories and impart new artificial mechanomemories. And within this big picture goal, we have some key questions. So we want to know how the spatial distribution of mechanosensitive factors, epigenetic modifiers enable mechanical memory. We want to know how temporal variations in epigenetic modifications, mechanosensitive gene expression can change rates of memory acquisition and degradation. And we want to know how mechanical changes then impact nuclear structure, chromatin spatial organization, and again, impart mechanical memories. And I'm going to give you some examples, particularly focusing on this last question of some of the research outcomes from the center. But before I get into that, I want to also highlight some of the tools that we're using to interrogate this. And so we are taking advantage of recent advancements and revolutions in sequencing technologies that allow us to not just sequence the linear code of the DNA, but actually understand its 3D and 4D folding inside the nucleus. And these include technologies like ATAC-seq that tell us about the accessibility of the underlying DNA, Hi-C, which tell us about the 3D folding and the contacts between different genomic regions. And we're taking these tools and we're applying them not just on cells grown in a glass or plastic petri dish, but on cells that are experiencing different mechanical environments, and in addition, changing mechanical forces. Additionally, we're taking advantage of recent advances in imaging technologies, in particular super resolution light microscopy that enables us to visualize the spatial organization of chromatin inside intact individual cells at nanoscale spatial resolution. So we can see these nanoscale domains that you already heard about a little bit from Ayush's talk. And finally, imaging and genomics are complementary. They give us complementary information. They have complementary advantages and disadvantages. And we would like to be able to integrate all that information together. And we're doing this through modeling. So we're developing predictive models that integrate information from sequencing and imaging to be able to predict how the spatial organization of chromatin changes in response to mechanical cues and how that then changes transcriptional outputs of specific genes within the nucleus. And so that's sort of a big introduction, and I'm going to go into and highlight a couple of examples of recent research advancements focused on this topic. And the first one that I'm going to highlight is a collaboration between my lab, Suchin Hio's lab, Rob Mock's lab, and Vivek Shenoy's lab, where we took advantage of advanced microscopy, super resolution microscopy, to visualize chromatin spatial organization in single tenocyte cells. So tenocytes are resident cells of the tendon. Tendon is a highly mechanoactive tissue. It connects bone to muscle. It's constantly under mechanical forces, and the resident cells, again, experience those forces and respond to them. And what we did in this work was to actually work with human tenocytes. So these are tenocytes that come from human patients. And we isolated either tenocytes from a healthy individual or an individual suffering from a disease called tendonitis, which is the inflammation of the tendon tissue that actually leads to changes in the mechanical properties of the tendon tissue. It, in particular, makes the tissue softer. And then we also looked at an aged individual. And what you're seeing here are, again, super resolution images of chromatin, color-coded in a way where blue represents more open, more euchromatic regions of chromatin, and red represents more condensed, more compacted regions of chromatin. If you look at an aged individual, we see more blue. So that suggests that there is less heterochromatin and more euchromatin. And this is expected, we know from other work, that in aging, a heterochromatin becomes disrupted. But if you look at the tenocytes coming from the tendonitis patient, we see something quite remarkable. We see a dramatic remodeling of chromatin such that it becomes more compacted, opposite of what's going on in aging. And it becomes more enriched at the nuclear lamina, which is known to be a silencing compartment. And so we wanted to understand some of the mechanisms that are mediating the spatial remodeling that we see in tendonitis. And again, we knew that the mechanical properties of the tissue changes. And so we took the tenocyte cells and we grew them on hydrogels of differing stiffness. And so we can make these hydrogels stiff or soft. And glass is sort of our control, which is the stiffest substrate. And I hope you can appreciate that if we put the cells on soft substrates, the chromatin spatial organization actually resembles what we see in tendonitis tenocytes. So these are tenocytes from healthy individuals, and we can modulate their chromatin by growing them on different stiffness substrates. So on soft substrates, chromatin becomes dramatically remodeled. And so the conclusions from this is that tenocytes from these tendonitis patients have aberrant chromatin organization, and that remodeling is partially mediated by substrate stiffness. And this was my favorite experiment. So we can do these experiments a little bit more dynamically, where we put cells on stiffening hydrogels. So these are hydrogels that are soft to begin with, and then we can stiffen them by cross-linking with light. And we did this experiment first with young and healthy tenocytes. And you can see that the chromatin spatial organization adapts to the changing environment. So on soft substrates, we see that aberrant remodeling. But as soon as we switch them to a stiff environment, within many hours, they spatially remodel their chromatin and have a chromatin organization that resembles more the healthy state. If we do the same experiment with diseased tenocytes, we no longer see that spatial remodeling and that adaptation to the new mechanical environment. So these cells have actually lost their mechanoepigenetic memory. And Vivek's lab helped us understand some of the mechanisms that are mediating this by developing, again, models. You heard quite a lot about this from Ayush already, so I'm going to keep it brief. But the key ingredient in this model is these rates of kinetics of methylation and acetylation reactions that either deposit repressive modification marks converting euchromatin to heterochromatin or vice versa. And using this model, what they found is that if we have increased repressive methylation modifications on soft substrates, then the model can fully recapitulate what we see on soft substrates, where chromatin becomes more condensed and more enriched at the nuclear lamina. And we were able to actually validate that there is increased repressive methylation in these cells on soft substrates. So where are we going with this? We're very interested in digging deeper into the mechanisms that lead to this spatial remodeling, and particularly the loss of mechanoepigenetic memory, because if we can understand that, we can potentially revert this, and this can have therapeutic implications. Now, in this work that I highlighted, we use microscopy, which is great because it gives us, a single cell information. But we were seeing this global remodeling of chromatin, but we were blind to what exact genomic loci are changing and what regions are changing. So the next work that I will highlight, again, a highly collaborative work from the center, particularly Annette Diamond and Rob Mock's labs, decided to take an alternative approach to look at this using genomics and sequencing to understand what are the genes that are changing. And to do this, they again use tendon. This is a mouse tendon model, where they take this flexor tendon and they resect it at a given time point. And that resection leads to loss of mechanical tension. So this is an acute detensioning model. What they showed is that upon resection, upon loss of that tension, the cells actually quickly respond. These are, again, resident, these are fibroblast cells, and they change their nuclear morphology in response to mechanical detensioning. And when they looked at gene expression using PCR, they saw quite a lot of changes in response to detensioning. In particular, they saw upregulation of matrix catabolic genes, which is quite remarkable to me because it means that the detensioning is leading to changes that then feed back into the matrix. So the matrix then becomes degraded further. And so there is this feedback between what's happening in the external environment, what's happening in the nucleus, and then that further impacts the extracellular matrix through production of these catabolic enzymes. Now, this was just looking at a few genes. They decided to take an unbiased approach to look genome-wide. And to do that, they developed a cell model of this detensioning where they used blebistatin treatment to lead to cytoskeletal detensioning. And then they did RNA sequencing as well as attack sequencing. And once again, they saw a huge response. A lot of genes went up and down. And again, they validated that the catabolic genes are going up upon detensioning. And what they saw in the attack seq was something quite interesting. They saw decreased accessibility of chromatin in response to mechanical detensioning, cytoskeletal detensioning. And this is in line with what we saw on soft substrates using storm microscopy, where cells have less contractility. And we also see a condensation of the chromatin and less accessibility. And this accessibility was particularly impacting target genes of the YAPTAZ pathway and mechanosensitive pathway. And so just to sort of sum this up, aberrant loss of tension leads to huge changes in the nucleus. It leads to decreased chromatin accessibility, decreased accessibility at the nuclear YAP targets, and increased matrix catabolic genes. And that further then degrades the extracellular matrix, leading to further changes in the external environment. So where are we going with this? So, they looked at these changes using these isolated cells from their mechanical environment and developing cell models. We would really be able to love to do this in the tissue context where we don't have to take the cells outside of their tissue context. We want to be able to visualize how, for example, resection or mechanical detensioning impacts the matrix, impacts the tissue structure, and how those changes lead to changes inside the nucleus. And to do that, we are using expansion microscopy, which is a super resolution microscopy approach that enables us to look at things at nanoscale resolution in intact tissues. And here I'm showing you an example of a mouse tail tendon where we were able to expand this tendon and label proteins and carbohydrates. So we can see, the collagen fibrils in the tissue. We can see the nuclei, and we can add on immunostaining to this to visualize the epigenetic marks in the nucleus, to visualize the chromatin within the nucleus, and we can add FISH to visualize gene activity. And so with this approach, we can study directly how changes in the external tissue environment impact cells that are directly adjacent to that microenvironment. So this is something that we're currently working on. And the final work I want to highlight is work from Becky Bell's lab in collaboration with Vivek's lab. When we talk about mechanical cues, we often talk about mechanical cues coming from the external environment, transmitted through the cytoskeleton. But Becky's lab showed very beautifully that internal organelles can also impact the nucleus and can also apply forces to the nucleus. So they did this in hepatocyte cells, and these are hepatocyte cells that have increased amounts of lipid droplets, which is something that can happen in metabolic diseases. And these lipid droplets cluster around the nucleus, and they can apply mechanical deformation to the nucleus. They can change chromatin condensation, and they can impair hepatocyte function. And this is quite remarkable to me. I think it opens up a lot of new avenues. My lab, for example, works on protein aggregation, in particular, aggregation of the protein tau in tauopathies and Alzheimer's disease. And we see a lot of tau inclusions around the nucleus. And I've been wondering ever since I saw this work, whether those inclusions can also impact the nucleus and change nuclear function. And so I think, we need to start thinking about how forces from the inside of the cell can impact the nucleus. And you already saw this from Ayush earlier, but again, Vivek lab was able to develop models that explain the changes that happen at the chromatin level when these lipid droplets are exerting forces on the nucleus. And finally, mechanics is not just important in animal cells. Our center is a cross-species center where we're also studying plant cells. And as you already heard, plant cells can experience mechanical forces, for example, through osmotic swelling or shrinking. In addition, pollen tubes grow in between cells in order to deliver sperm to the nuclei for fertilization. And so they experience mechanical forces. And all the tools that we have been developing in animal cells are also applicable to studying how those forces change the nuclei in plant cells. You already heard about these tobacco cells expressing histone H2B fused to YFP. And so the Anderson lab and the HEO lab have a seed grant from CEMB where they will be using these tobacco cells that are labeled and advanced microscopy, including super resolution light microscopy, to study how forces, for example, osmotic forces, impact nuclear organization in these plant cells. And then just to sort of wrap everything up, the impact of the work that I've been telling you about is to engineer chromatin organization and genetic programs using mechanical stimuli, both in plants and animals. And if we can do that, there will be quite interesting applications that are therapeutic as well as agricultural. And so that's sort of the big picture goal of what we hope to achieve. And I was only able to highlight just a couple of different examples in my talk of research done on this topic, but we have huge amount of work that is being done collaboratively. And so I highly encourage you to go out and check out posters that will be today on this topic, particularly from the HEO lab. We have several poster presentations on using advanced microscopy, including super resolution microscopy and single molecule tracking to look at how forces impact nuclei of different cell models. And several posters from the Shenoy lab taking both a data-driven as well as a more mathematical approach to model how the nucleus changes in response to these forces. And with that, I'm happy to take any questions you may have.
Wonderful talk. I was quite interested in your photo stiffening gel experiment where you saw, sort of broad changes in chromatin compaction upon stiffening. Have you had a chance to do sequencing or other analysis in that context, in that paradigm as yet? And is it your sense that there are specific loci that are particularly stiffness responsive?
Yeah, wonderful question. And I don't know where Suchin is, but his lab is currently doing some of those experiments. So we do think that it is impacting tenocyte-specific genes and then impacting tenocyte phenotype and function. And so we are gearing up to do these sequencing experiments. They require us to grow a lot of cells. So that's one of the limitations. But this is exactly what we want to do next. Yeah.
I also had a question about that chromatin reorganization upon stiffening, and I wonder how much in that system chromatin organization changes are coupled to nuclear shape changes, because as cells adhere, their nuclei flatten. So I wonder how much you've looked in 3D, and also apical versus basal, because there's some indications that at least the lamina changes its conformation on the basal versus the apical surface in response to that tension.
Those are excellent questions. So in terms of cell volume, we did not see huge changes in cell volume, but we didn't look at sort of the morphology. So that would be something interesting to look at. Basal versus apical, we're sort of taking a midsection through the nucleus, so we didn't look at those differences. But what you mentioned sounds incredibly interesting. So yeah, something to think about.
Thank you. I couldn't help but asking about your wonderful work. A tendon binds to a hard surface and a soft surface with the same cells. And I'm just wondering what differences you might see from the interface on it. You have to have a gradient of rigidity, it seems to me, for the extracellular matrix. But do you see a difference in the cells at either end of the tendon when you and if it's a normal tendon?
This in the tissue context, because, you know, when we isolate the cells, we don't know where within the tissue they came from. And so we can't really address those type of questions that relate to the spatial organization within the tendon itself. So the expansion microscopy work that we are doing currently, which is still very preliminary, and we're even amazed that we can expand these very rigid tendon tissues, but that will go on to address some of those type of questions. So stay tuned.
Thank you. Thank you, Dr. Lakadamyali.